Meiosis is important because without it, there's no variation. And without variation, there is no natural selection. And the sources of variation are crossing over, where you end up with chromosomes that are part maternal, part paternal, as we've talked about in the past. So you end up with a chromosome that has crossing over occurring. Independent assortment of chromosomes, which we haven't hit on too hard yet. I'll get there in a second. And mutation, that's when you used to have ACC and now you have ACG. You've had an error, a change during S phase of the cell cycle. You've put in a different base. You've changed the protein that will be produced. That is another source of variation. There's one other one that's not on the list, which is kind of the random nature of fertilization, whereas you'll have like one egg and 2 million sperm trying to fertilize that egg, and each sperm's a little bit different. So that's a source of variation. So looking at this in this form, here we have a mom and a dad at the top, and let's say this is chromosome number 1. Dad has 2 copies, so he has copy 1 and copy 2. They're homologous, right? Mom has copy 1 and copy 2, and those two are homologous. Dad only puts one of them into a sperm, so let's say it's this one, goes into a sperm. Mom only puts one of these into an egg, let's say it's this one. Okay, so the alleles, or the chromosomes in this case, segregate, right? Now they fuse together into this new person right here who has a copy of her dad's chromosome and a copy of her mom's chromosome. Now when she goes to make a gamete, those two are going to undergo crossing over, and so her egg is going to have recombinant chromosomes that are part of her dad's chromosome and part of her mom's chromosome stuck together. So this part is paternal or maternal, this part's paternal, right? So she took these 2 chromosomes and she mixed them up. So that's why meiosis is so important. You get crossing over occurring, which gives you chromosomes that have parts of your dad's chromosomes attached to parts of your mom's chromosomes. So we don't get new alleles here, but we get new combinations of alleles here. And I just realized I don't have my light on. I'm going to turn it on here to help you see me a little bit better, as if it really matters if you can see me. So this is how we get new combinations of alleles. So you might have your mom's hair color and your dad's eye color on the same chromosome now. Now, independent assortment of chromosomes is a different thing entirely. This is how chromosomes line up on the metaphase plate. So let's say, you have chromosomes lined up in metaphase 1. That's where this takes place, how they line up. So in this example, we have a cell of 2n equals 2. I'm sorry, 2n equals 4. And the metaphase plate's up and down here. It's kind of hard for me to draw real accurately, but there's the metaphase plate right between those. Possibility 1, all the paternal chromosomes are on the left and all the maternal chromosomes are on the right. Possibility 2, we have a paternal over here and a maternal over here, but we have a maternal on the left here and a paternal on the right. So they flip places. So think of it this way. For every chromosome pair that lines up, they could line up this way or this way. For every single pair. That's independent assortment. There's no it's random which way they line up. The paternal one could be on the left in this case, or you could think of it up and down, or it could be the maternal one. It's random. But it has big consequences. So we're not counting crossing over here because that would complicate the issue. But here we see these chromosomes. Here we see them, the outcomes of meiosis 1 and then meiosis 2. In this case, we end up with gametes that are all paternal and all maternal. So we end up with 4 gametes that are either all paternal or all maternal. And once again, we're not looking at crossing over on this image. In this example over here, possibility number 2, we end up with gametes that are recombinant. They have they have paternal and maternal chromosomes in them. And that's because of independent assortment. So let's look at another example. Here's an example of a cell of 2n equals 6. And here are all the different ways that maternal and paternal chromosomes could line up. So here's combination number 1, all paternal on the left, all maternal on the right. Combination 2 is just the mirror image of that. But then there's all these other different ways they could line up. And the way to calculate how many possibilities there are for this is to take 2 to the haploid power. So for example, 2n equals 6, so n equals 3, so 2 to the third power is how many different combinations you could get through independent assortment. So 2 times 2 times 2. So 2 times 2 is 4 times 2 is 8, and you'll notice there's 8 different combinations shown here. For human beings are 2n equals 46, so n equals 23, so 23. So 2 to the 23rd power is how many different combinations of chromosomes are possible without crossing over, just with independent assortment. Whether the paternal chromosome's on the left or the right for every single pair. So for chromosome 1, it could be this way. For chromosome 2, it could be that way. For chromosome 3, might be back this way again. So it varies from chromosome pair to pair which way they line up, and that's a source of variation. And that will affect the gamete. So this is over 8 million combinations in human beings, and that's not even taking into account crossing over. Okay, so at what stage is this determined? This could be a test question. When does independent assortment take place? That's going to take place in metaphase 1 when they line up at that metaphase plate. So looking at this again, we have prophase 1, chromosomes line up, crossing over occurs, independent assortment occurs, then we divide things out and into metaphase 2, and at the end of metaphase 2 we have 4 gametes produced. Some of them have chromosomes that are recombinant, part paternal, part maternal. And so that's one source of variation. The other source, of course, is independent assortment. Now, there can be errors in meiosis that take place. I want to mention that. And one of the major sources of error is known as non-disjunction. That is when a chromosome fails to separate. So, for example, rather than that separating out into this and this, maybe they go together, okay? Or rather than these two separating apart from one another in meiosis 1, they are inherited together. And this is how you end up with extra chromosomes or too few chromosomes. So you can end up with something like Down syndrome here. So this is important because if you have an error in a sperm or egg, so let's say you have a sperm and it's supposed to have 1 chromosome in it in this example, but instead it has 2, and then it fertilizes an egg, every cell of this zygote that's going to be produced is going to have extra copies of that chromosome. And so when this divides by mitosis, it's going to make copies, right? So every cell produced by this fertilization event has that extra chromosome in it. So every cell of the new organism has an extra chromosome. So here's an example. Down syndrome is caused by trisomy 21. So here's a karyotype. Once again, a karyotype is when you have an image showing all the chromosomes in a cell. And we can see that someone with Down syndrome has an extra chromosome number 21. And that results from non-disjunction during meiosis. In this case, we have a female with trisomy 21 showing probably the Down syndrome phenotype. Okay, so these questions you can look at on the PowerPoint version of this that I have posted. But those are some of the ways that variation arises from meiosis and what happens when it goes wrong. Thank you for joining me, and I hope the sound works on this video, because it didn't the first time I recorded it. All right, let me get out of here.